Okay, let me introduce today's speaker. Today, Irene Chen from UCLA, Department of Chemical and Biomolecular Engineering is actually going to give a talk. She received her PhD in biophysics from Harvard in 2007, and she followed by the postdoctoral research as a Bauer Fellow in systems biology at Harvard University as well. And then she actually became an assistant professor in 2013 at UC Santa Barbara, and she stayed there for like about 7 or 8 years. And then she last year she moved to UCLA in the current position. And today she's going to talk about the emergent byproducts of evolution. Okay, please welcome.
Thank you. Thanks for inviting me to this virtual summer school. I have a few different topics I want to try to talk about. A couple of them are directly related to the origin of life or emergence of life. And then one of them is a little bit more applied. And I hope you'll see the connections between the more basic science projects that we've been doing and the applications. So just to give you a little outline, the talk is kind of divided into 3 parts. So the first part, I'll talk about the RNA world and evolution in the RNA world and the concept of frustration and frustrated evolutionary networks. And then I'd like to talk about environmental effects that can lead to emergent properties. And I'll be focusing on vesicles and what vesicles could do for RNA and RNA evolution. And then the last topic I want to cover is using evolved robustness in biomolecular engineering. And this is something that started out as kind of a separate line of research, but I think some of the lessons that we've learned have kind of dovetailed with our thinking about molecular evolution. So after each of these parts, I'll try to pause and give people an opportunity to ask questions if they'd like to ask questions. Since it's a pretty long period of time, we would kind of break it up that way. Okay, so I think by now you've probably already been introduced to the idea of an RNA world, and perhaps you've even seen this diagram before. This was originally from a review article that Jerry Joyce wrote in 2002. And since then, it's kind of undergone some additional modifications, but I believe this is the original version. This is a rough timeline of the emergence of life, starting with the formation of the Earth, going through a prebiotic chemistry kind of generation of these small molecules that are needed to feed the earliest life. The polymerization of these small molecules into information carrying molecules, the RNA world, maybe or maybe not a pre-RNA world. We won't really talk about that. So in my lab, we kind of take the existence of these polymers for granted. So we assume that there's some RNA in there. And in the timeline of life, the RNA world then transitioned to the DNA, RNA, protein life. And the major innovation here was protein translation, which I'm going to talk more about. And then, of course, this early life diversified into all the different forms that we have now. And the RNA world is, I think, very fascinating because it marks the beginning of natural selection and evolution, genetic information. So that's a place where we can start to understand and try to probe evolutionary concepts. So the RNA world idea was developed in 1967 by Francis Crick, Leslie Orgel, and Carl Woese. So that's, a long time ago, basically. And they had this idea that RNA could act as both genome and biocatalysts. And I think the reason why it's fascinating that the idea was developed so long ago is because this idea actually predated the discovery of catalytic RNA. So the feature that these 3 biochemists were looking at was the observation that RNA in tRNA could fold into very complicated structures. And to them, it looked about as complicated as a protein structure. And so they thought, well, if RNA can adopt these kinds of folds, and folding is it is what determines function, then perhaps RNA can have function and the same functions that proteins are known to have. And then in the 1980s, we got some evidence from biology with Tomczyk and Sidney Altman discovering ribozymes, catalytic RNA. Then there's also this observation from probably anybody who has looked at a biochemistry textbook, is that it's very complicated, and perhaps it looks even more complicated than it needs to be because you see little bits of RNA in various places like ATP and ADH, and it's been argued that there isn't an obvious reason for why there needs to be these sort of RNA handles on metabolism. So that's thought to be maybe a historical artifact. And then the third piece of reasoning that people point to for the RNA world, or the existence of an RNA world, is the structure of the ribosome. I know this is a varied audience, but the ribosome is the protein RNA machinery that translates mRNA information into protein sequences. So it's like the decoding machine. And the ribosome itself was known to consist of RNA and protein. And when the structure was solved in 2000, it was realized that the catalytic core of the ribosome, which is shown here, the catalytic core is actually completely made of RNA. And all the protein, which is shown in these kind of like bluish purple structures, the protein is pretty far away. And there's no way that the protein is directly affecting, or I should say directly participating in the catalytic mechanism of the ribosome. So the interpretation of this was that the ribosome is a ribozyme, which has been stabilized and elaborated by proteins. And then following from that, that's taken as the smoking gun, circumstantial evidence that there was an RNA world, because the machine that makes proteins is at its heart an RNA. Okay, and I also like to sometimes just reflect on how interesting it is that RNA has this dual function of simultaneously carrying information and encoding and having function. And I think it's analogous to what's depicted here, where suppose you wrote a paragraph about a horse, and the words, the information itself could function as a horse. And that would be pretty amazing. So I think it's pretty amazing that RNA is able to have this, this kind of property. Okay, so I think the RNA world is it's been called very elegant. It's very cool as an idea, but at the same time, it's not very practical to try to convince anybody who's skeptical that there actually was an RNA world. What I think that we can all agree on, regardless of how skeptical you are about the kind of history of life and whether or not there was an RNA world, what we can agree on, I think, is that the RNA world is a good model system for studying how life can emerge. So if we can create in the lab something that resembles an RNA world, it is much simpler. It will be much simpler than a DNA, RNA, protein world. So it has the potential to be a stepping stone between an abiotic system and something that we would all recognize as being alive. So in practical terms, just a very utilitarian perspective is that regardless of whether it actually existed, some system that's based on catalytic RNA greatly simplifies what we would need to build an artificial living system, and therefore it's an interesting model for studying the origin of life. And some people might consider this bottom-up synthetic biology, this kind of trying to create these artificial cells. Okay, so where do these functional RNAs come from? They come from the process of evolution, either evolution in nature, as is the case with the first discovered ribozymes, or evolution in the lab, where in vitro evolution has given us many different functional RNAs of catalyzing different chemical reactions. One of my favorite examples is the RNA Diels-Alder-race. It catalyzes the Diels-Alder reaction, which was actually discovered before a protein Diels-Alder-race was engineered. So perhaps there are opportunities for stretching RNA catalysis in ways that proteins may be less amenable to. But that's just a thought. Okay, so evolution can be thought of as a walk over what's called the fitness landscape. And the fitness landscape is what you would get if you knew the fitness or the activity, let's say, of every single possible sequence in sequence space. So let's imagine like the space of all possible sequences. We kind of put them all down on a sequence coordinate in some order here so that the more closely related sequences are together. In reality, I guess, sequence space is multidimensional, but we'll just represent it this way. So the landscape is this function of fitness or activity. We'll be talking about activity since that's what we can measure for our RNAs or ribozymes. The function of fitness over the sequence space. So just to look at this for 1 second, these peaks in this landscape correspond to families of sequences, so related sequences, a group of related sequences that might perform some catalytic activity. And these lower lying areas are sequences that are inactive or less active. So if we knew what this fitness landscape looked like, if we had, a map of this landscape, then you could infer some things about evolution, like how many different peaks are there, how many different solutions are there to this catalytic problem, and how difficult is it to start out from a low-lying area and discover a higher peak. And then also, what are the chances that I'll discover a suboptimal peak versus the globally optimum peak? Is it possible to get from here to here, or will you just end up on these kind of suboptimal peaks? So to answer questions like that, what we'd really like to have is a map, an empirical map of this fitness landscape. That's typically basically impossible due to the large number of sequences. 4 to the n possible sequences gets out of control very quickly. But ribozymes can be quite small. So if we focus our attention on small RNAs, like a random region of 20 to 30 bases, you can get something functional out of something that's that small. And you can also probe the entire sequence space or almost the entire sequence space through a random library. So to try to carry this out, our approach is based on in vitro selection. So we start out with a library of 4 to the n, and n was 21 in this case. So we have 21 randomized nucleotides, and we have some flanking regions for PCR for technical reasons. And in this random library, we basically have maybe roughly 1,000 or a few thousand copies of each possible sequence. It's stochastic, so we don't necessarily have like 100%, but we have probably 99.999 something percent of sequences represented in there. So every sequence in this random pool is more or less at kind of an equal concentration in the beginning of the experiment in this random pool. And the idea is we're going to take some, we're going to apply some selection, some selective pressure to remove sequences that are inactive and try to keep the sequences that are active. And by doing this selection, we'll be able to rediscover the form of the fitness landscape.
I have one quick question. So
yeah,
when you say the activity, how do you actually judge the activity? Catalytic activity in terms of activity described by Michaelis-Menten type of reaction rate?
Okay, thanks. So I'll talk about the reaction that we're looking at. One could probably do this for different types of reactions, and we limited ourselves to one specific reaction. If we're trying to, in our case, it's described by a single reaction parameter, so a kcat. But you could imagine more complex reactions. You could also think of different features that might play into fitness, like stability or other properties of the RNA, how good it is at being a template and that kind of thing. We have focused on activity. So I guess that I should probably more correctly call our maps activity landscapes instead of fitness landscapes. Okay, so that's a great segue. What are the ribozymes? What's the activity that we're interested in? So we've been very curious about the genetic code. People have called the genetic code a major evolutionary transition, and certainly it was a transition out of an RNA world and into a protein-based world, the way that we have the central dogma now. So that does seem like a really a critical transition. You can kind of think of the genetic code in two parts. There's one part which is kind of the connection between specific amino acids and specific codons. Sometimes people call it the second genetic code, but that that mapping of codons to amino acids is performed by a system of enzymes called the aminoacyl tRNA synthetases. So these aminoacyl tRNA synthetases attach specific amino acids to specific tRNAs, and those specific tRNAs have the right anticodons so that they will match up to the codon. And then the second part of the protein translation is the ribosome and all of the functions that the ribosome performs. But we thought we'd just kind of focus in on this so-called second genetic code, the matching up of the amino acids to the tRNAs. So it's been shown that ribozymes can perform this function. It's been, I guess, many years, a couple decades now, that people have known that ribozymes can perform this same reaction. So some of the technical features of the previous selections have been, for example, using the first one in Michael Yaris's group, used an aminoacyl adenylate, and that's highly reactive in water, pretty difficult to work with. Not particularly prebiotically plausible. So we were looking for alternatives. This is an interesting overlap where something that is prebiotically plausible, it also tends to be something that's easy to work with in the lab. So aminoacyl adenylates are reactive, unstable in the aminoacyl tRNA synthetases. These enzymes kind of shield the adenylates from water to prevent them from hydrolyzing during the reaction. So in this case, the fact is that they're not prebiotically plausible, and then also they're highly reactive and difficult to work with. So a nice place to go if you're looking for something that might be easier to work with in the lab is something that it also happens to coincide with something that's prebiotically plausible. And I'll introduce that on the next slide. And a second consideration here is that if we're curious about using these ribozymes in synthetic cells, then perhaps this particular, the adenylates, are not ideal. Now, there has been a very nice series of work by Hiroaki Suga's group, and they've been looking at the flexozyme for charging tRNAs in synthetic biology. So this ribozyme activity has been found more than once, and we decided to look at this from the prebiotic angle and use a substrate that was relatively stable in the lab. And that is the 5,4-H oxazolones, and that structure is shown here. This, you can kind of see the backbone of amino acids. Here's the amine, here's the carboxylic acid. This R1 is going to be the side chain. And this R3 is a place where you can attach something, and it was very convenient for us to attach a biotin, which we could then use during our selection, which I'll explain, for pulling down reactive molecules. So these 5,4-H oxazolones are produced alongside with N-carboxy anhydrides, NCAs, which are widely considered to be prebiotically plausible activated amino acids. These NCAs definitely condense and polymerize. They're produced in simulations of volcanic gases. What's very nice is that Ziwei Liu, our collaborator, who was working with Robert Pascal when he started to do this, and then moved to John Sutherland's group after his PhD. What Ziwei realized or observed was that if he mixed the 5,4-H oxazolones, and in here, this is a tyrosine analog, it's a O-methyl tyrosine, then they react slowly with nucleotides. So the nucleotides have the OH nucleophile, and they do slowly react to form this product. So what's nice about this for us is that it means the reaction is thermodynamically downhill. It's occurring at a slow rate. And so we just need to select an RNA that will accelerate this reaction. So our job here was to take a random DNA pool, transcribe it. So now we have a random RNA pool. React it with the oxazolone substrate. Here's the side chain tyrosine analog, O-methyl tyrosine, and biotin. And if there's a reaction of any of these molecules with the oxazolone, then that will have the effect of biotinylating the RNA. So then we can isolate these reacted RNAs by incubation with streptavidin beads. This will pull down the reacted RNAs. We can amplify them by RT-PCR and repeat this cycle to enrich the reacted RNAs and follow the progress of this selection by high throughput sequencing. So I won't go into all the details there because I'd like to focus on a few other things, but this selection did yield a few different families of ribozymes, and I'm showing the structure here, or I mean the sequences here. 3 different motifs, and there are a couple of subfamilies within each motif. And this is the reaction that they were observed to carry out. At this point, what we wanted to do was to associate a number with each of these sequences. So in the selection, we basically use that selection to like throw away all of the inactive sequences, and then we still keep the active sequences. One way to try to get some fitness metric would be to just say that the abundance of the sequence after the selection is kind of the fitness. But we wanted to actually get a activity metric like a KCAT. So in order to do that, we took a pool of these sequences, not completely random because that is too complex to analyze, and also not too converged because then we only have a few sequences there, but something with maybe like some thousands or tens of thousands of sequences in it. And so we take that pool, a kind of a medium diversity pool, aliquot it into different concentrations. So we have a concentration series. You could also imagine doing this as a time series, a series of time points. And then, so we have triplicates at each different concentration of substrate. And then we separate the reacted and unreacted fractions using the streptavidin pull down, and we sequence the reacted fraction. And then if we have an absolute quantitation of how many sequences were in the RNA that was pulled down, we can get an absolute quantitation of how much RNA was reacting at the different concentrations. So that will allow us to fit the points. In this case, I think we had 4 or 5 different concentrations. We can fit those to a kinetic model. And the appropriate kinetic model for us was a first order model. So we can see that this measurement, which we're calling kinetic sequencing or KSeq, this measurement lines up pretty well with what a traditional biochemical measurement would be, a gel shift assay, where you mix your single sequence. So take, one sequence by sequence, take one sequence at a time, mix it with, react it, and then mix that with streptavidin and run it onto a gel so that the reacted sequences, that fraction of sequences will migrate more slowly, so you can calculate how much was reacted in the gel shift assay. So we get a pretty good alignment of these values. So now we have basically a long list of maybe thousands of different ribozyme sequences that fall into a few different families. And our question was to go back to our kind of initial thinking about fitness landscapes and what we could analyze if we knew if we knew the entire structure of the landscape. So one of the quotes that inspired us, some of the work that inspired us here, is this idea from John Maynard Smith. If evolution by natural selection is to occur, functional proteins, RNA sequences, in our case, must form a continuous network which can be traversed by unit mutational steps without passing through non-functional intermediates. So what he's saying is that if you want it to have an evolutionarily accessible pathway between one sequence and a different sequence, then it has to be possible to find a path where you are making mutations one at a time, more or less one at a time. And that path does not have a member that is so low in fitness that the population can't survive going through that low fitness intermediate. So some very nice theoretical work had been done on this on the computational side. This is all before high throughput sequencing and the ability to gather this information. So during that time, Peter Schuster's group had worked on folding RNA secondary structures, where prediction is not too bad. I think the numbers are something like 80% accuracy in prediction of base pairs. So if you take these RNA folding algorithms and predict the secondary structure of different RNA sequences with thousands or tens of thousands of these sequences, and you ask the question, if I have a sequence A and it folds into a particular secondary structure, and I have a sequence B that folds into the same secondary structure, is it possible to find a series of mutations that changes A into B, all the while preserving this secondary structure? And the answer to this question was actually yes. Their work showed that for most of the common structures, you could find these so-called neutral pathways, which gave this neutral network through sequence space. And that kind of gives rise to the idea that if there is a neutral network through sequence space, then you could arrive anywhere in sequence space from any starting point, just traveling neutrally without any fitness decrement or without a terrible fitness loss. So we decided to see whether this translated into real life, I guess, into this catalytic activity that we were measuring. And what we found is that here's an example of a pathway that we looked for, going from sequence 1B, sequences are given along the side here, through sequence 1A, over to the global optimum, which was sequence 2.1, and a lesser minor relative 2.2 here. So what you could see is that for the sequences that were recognizably in the same family, there are pathways that can connect these sequences, and fitness doesn't drop off too greatly while going through this pathway. But as soon as you need to jump to a different motif, let's say you wanted to find the global optimum here, if you were hoping, starting in this kind of lower activity motif, you're hoping to find this high activity sequence, there are many steps required at low, essentially zero fitness in order to cross from this kind of mountain number 1 to mountain number 2. So only nearby peaks, these kind of like locally connected peaks, only between those do we see the viable evolutionary pathways, but between these like major changes in motifs, we don't see them. I think that makes a lot of sense, but it just points out the contrast with what was observed in the theoretical simulations, which were based on secondary structure prediction. So that was a single evolutionary pathway. And if we kind of zoom out to look at the network in a little more detail, then we can see here sequence 2.1. This is a heat map of the catalytic activity. Sequence 2.1, again, was the global optimum of this landscape. It has this relative 2.2. There's an isolated motif here, 3.1. What's shown here are individual sequences. So each dot corresponds to an individual sequence. And the type of line that is connecting two sequences shows how many mutations you would need to go from one sequence to the other sequence. So 2.1 is connected to 2.2, at least through this kind of bridge, although one of the sequences is fairly low activity. But it is not connected to motif 3.1 or to this larger kind of plateau of what we call motif 1, which encompasses a lot of different sequences. So if you are so lucky as to start out close to motif 2, you can indeed climb to the optimum. But if you are unlucky and you start out closer to motif 3 or motif 1, maybe more likely motif 1, then it would be difficult under selection to traverse the path from motif 1 to motif 2.
So is this all of the structures have the same secondary structure?
No, that is probably not the case. So in this case, they perform the same function, but they do not have the same secondary structure. So that's where the analogy kind of breaks down there. Probably motif 1b.1 and motif 1a.1 have related secondary structures, although not identical. But between motif 2 and motif 1, we don't really see any resemblance.
Okay. And a single mutation, if you preserve in the secondary structure, that means your single mutation is made not in the stem, actually. It may be made in the stem loop or some bulge and things like that, no?
Yeah. So actually, I didn't bring a slide with this, but if we align the sequences like from this area, 1b.1, 1c.1, 1a.1, if we align those to each other, the major differences between these kind of clusters are changes in the flanking regions. So we see kind of the core motif moving, kind of sliding within the larger 21-mer sequence.
I also, I might have missed this, but what is the function here that's being defined if it's not structured?
It's self-amino-acylating RNAs. So they react with an oxazolone to
Sorry,
produce...
I mean like between like 1 and 2, B and C, etc.
Oh, what do these numbers mean?
Yeah, how do they, is it, I thought you said they perform some sort of different function, these different regions?
They're, I guess, all performing the same function in that they're all reacting with the substrate, the same substrate, but they all have different secondary structures. So if we, at least if we fold them in silico, okay, all of these 2-point-somethings fold differently from the 1-point-somethings, which fold differently from the
a region that's connected by at a distance of 1 is roughly the same structure, I guess.
Yes. Okay. Yeah. Okay. So I guess that when we looked at this, we thought about the distinction between what this kind of network looked like and the neutral networks that were found in the secondary structure work, and we like to say that this network, in contrast to the neutral networks, is kind of a frustrated network because it's difficult to optimize function over this network. Only if you happen to be close to sequence 2.1, then you can kind of get to the optimum. Otherwise, it's very likely that you'll be trapped on a suboptimal peak or, many different mutations at low fitness would be required to get to the true optimum of the landscape. So for this audience, I thought it'd be fun to just point out the analogy to frustration in some other systems. In protein folding, this concept is used to describe the difference between kind of a very nice folding landscape where no matter where you start in conformational space, the protein is funneled into the minimum energy fold. Compared to a frustrated landscape where maybe you get a lot of different misfolded forms that are local minima. It might be difficult to get into the native fold. And then there's this classic example of frustration, which is the antiferromagnet, where energy is minimized if you have plus spins next to, or up spins next to down spins. And that can be done in certain geometries, like in this kind of a checkerboard pattern. You can get ups next to downs, and the energy is possible to minimize. But if you have something like this, you have too many constraints where, like if you look at this one, for example, it's next to a down here, but what do you put here that won't clash with one of these? So just this geometrical arrangement causes frustration. So these systems are difficult to optimize in energy or in fitness or activity. And small perturbations in the initial conditions, like exactly where you start out on this conformational landscape, or on the sequence space in the space of all possible sequences, those differences in initial conditions lead to large differences in the outcome, exactly which of these minima you wind up in. And then one of my kind of fun examples of frustration is from this XKCD cartoon, which is illustrating what happens if you have people trying to sit down in a movie theater. They all have different interactions with each other. Some of them are friends, some of them are acquaintances, some of them are in a relationship, one-way crush. So this is too many constraints for solving the problem of everybody sitting down in a line in the movie theater. Here are two people in a relationship that this guy is stuck in between. Guys, this is not socially optimal. And maybe next year we'll be able to experience this in real life again. Okay, so that's kind of what I want to say about fitness landscapes and this property of frustration in the network. Are there any questions at this point before I move on to talk about environmental effects?
I had a quick point. I was wondering what would happen to the fitness landscape if you had added, for example, some sort of RNA chaperone into the mix?
That's an interesting question. So perhaps that would improve the folding. You could imagine that would increase the number of sequences that would fold into the functional form. Whether that could actually bridge the difference, the gap between, say, motif 2 and motif 1, I don't know about that. We know that the reactive sites are different, so they're presumably mechanistically distinct. So whether that could actually create connections between really different motifs, I think that would be difficult. But actually, the next project I'm going to, in this next section, I am going to tell you about an effect that is similar to chaperoning that we observe for RNA. And it does lead to some interesting outcomes. So maybe we could talk about that and continue.
All right, cheers.
Okay, so I'm going to talk about two different environmental effects. One is UV radiation, and that's kind of a prelude to what I guess I really want to talk about, which is encapsulation by vesicles. And like I was just saying in response to the question, the encapsulation of RNAs gives some interesting chaperoning effects. Okay, but I would like to start out talking about the UV radiation. So we got into this not because we thought it would give some emergent property or anything like that, but mostly because it was a fact of life on the early Earth. UV radiation is certainly damaging for nucleic acids. As we know, we put on sunscreens so we don't get sunburns, so that we don't get melanoma. But it's also UV light. The energy of UV light is also important for chemical synthesis. And there are several steps, for example, in this synthetic pathway that has been developed for describing nucleotide synthesis and also synthesis of some other important monomers. UV radiation appears to be quite important for that type of synthesis. So we'd like to understand what is the impact of UV radiation given that we kind of need it for synthesis and as a way to deliver electrons or to give us electronic excitation, but at the same time, it's detrimental to RNA. So we wanted to look into this a little bit. So thinking about what happens when a molecule is exposed to UV. So we have some energy landscape for this molecule. It gets excited. It's in the excited state. If it releases a photon, that's fluorescence, and goes back to the ground state. It might react, go off onto some other reaction pathway. That's considered damage because that could result in cross-linking of the nucleobases, for example. Or it could undergo, and the other way to dissipate this energy is through a process called internal conversion. And this is through some molecular flexion that happens to get the molecule into a conformation where the energy landscape of the excited state intersects with the energy landscape of the ground state. So the molecule can then re-enter the ground state. So these are so-called conical intersections, and they're out of equilibrium. They're out of the equilibrium geometry. So it requires some kind of flexion in the molecule. It's sensitive to the molecular context, to the RNA sequence, as we'll see. So it's kind of hard to predict these conical intersections. And so we just decided, let's take some RNA, expose it to UV, and we'll take some functional RNA, expose it to UV, and see what sites are damaged. So we chose a functional RNA called the Malachite green aptamer. So this is an RNA that binds to a small molecule called malachite green. And malachite green is normally not fluorescent, but if it's bound and its conformational freedom is restricted, then it fluoresces. So the activity of this aptamer can be followed by fluorescence, which is very convenient. So we expose this aptamer to UV light and then look at its fluorescence over time. And we can see that exposure to UV results in loss of activity. So this is somewhat expected because we know that we're damaging the RNA. And one thing that we can also see is that the binding pocket appears to be particularly destabilized compared to the global fold of the RNA. So here we're following on the left the melting of the binding pocket. So this is monitoring fluorescence as we increase the temperature. So without UV, we have a particular melting curve. But after UV exposure, then this melting curve is shifted substantially to the left. And so the binding pocket has been destabilized. And then on the other hand, if we just monitor by circular dichroism, we can monitor the overall fold, the secondary structure of the RNA. Then if we look at the non-irradiated versus irradiated samples, the melting point is actually fairly similar. It's more similar for this RNA measure when it's measured by circular dichroism than when it's measured by fluorescence. Indicating that the binding pocket is more destabilized. So then we'd like to know where exactly these lesions are on the RNA. And we make use of the fact that reverse transcription stalls at lesions. So if the lesion is encountered by the enzyme, then that causes stalling. So we can start with a template which is being copied by reverse transcription into the complementary DNA. If it goes all the way to the end, then it's a full length, and that's what you see if there's no UV exposure. You get full length reverse transcript. But if there is increasing amounts of UV exposure, then you get stalling, you get buildup of these shorter products, and that indicates that there's a lesion at this point. So in this way, we can follow the appearance of these lesions and the locations. And here, the area of the circle is proportional to the rate of damage accumulation for each of these different sites. So here's the structure of malachite green aptamer kind of written out schematically. And here's the binding pocket. This red cross is the malachite green, the position of malachite green. And what you can see is that the binding pocket is kind of disproportionately affected by irradiation. So we do see some patterns which are not too surprising, that consecutive pyrimidines are susceptible to damage, and that is known to result in pyrimidine dimers, like thymine dimers in DNA. We have the same phenomenon in RNA. And also these single-stranded regions, the binding pocket happen to be more quickly damaged. So as you can see from here, it would be hard for me to predict which of these positions would be more likely to suffer a lesion. So I guess to me, I consider this hard to predict. We looked at another aptomer also. So in here, blue is the malachite green aptomer. We also looked at a different fluorescent aptomer, spinach 2. It binds to a different dye, but is also fluorescent. And we can see that its activity, it loses activity at a different pace from malachite green. Single-stranded regions appear to be preferentially damaged, and UV hardiness varies among different sequences. So from this, I guess the kind of very bird's eye view picture of this is that the environment is an important influence. If you are comparing, if these two sequences were competing against each other in a UV-rich environment, then perhaps an advantage would accrue to the spinach 2 aptomer because it appears to be more resistant, or at least its activity appears to be more resistant to damage. Okay, so the second environment I want to talk about is vesicles. I'm sure that protocells have been introduced to you already. Vesicles are important for several reasons, and I'll just reiterate a couple of them. One is that it's a semi-permeable membrane that traps macromolecules. So RNA sequences, that information is encapsulated, while small molecules, mononucleotides or other kind of nutrient molecules or waste molecules, I guess you could say, those can permeate through the membrane. And the other function of having this membrane is, in evolutionary terms, it prevents parasitic sequences from overwhelming replication and metabolism. So keeping related sequences together prevents parasites that arise spontaneously in one compartment from kind of infecting all of the other compartments. So vesicles seem to be an important element of early evolution. They're also quite easy to form. But the environment inside a vesicle is not the same as the environment in bulk solution. And we know that from a lot of cell biology work and also from model studies. So we'd like to understand how the environment of being inside a vesicle affects the RNA. So the system we're using is primarily based on fatty acids. Fatty acids like myristoylic acid are this acyl chain with the carboxylic acid. And they have been found on meteorites where the composition of those meteorites is thought to reflect the composition of the early Earth. These carbonaceous chondrite meteorites, Murchison is kind of the famous one. So these fatty acids are found in this meteoritic samples, and they're also it's also possible to synthesize them abiotically. So they're thought to be kind of a prebiotically interesting model system. They could also have some interesting dynamic behaviors, which I won't go into, with growing and dividing and kind of performing some of the functions of a cell cycle. So we're using these large unilamellar vesicles prepared by extrusion, so you can get them fairly homogeneous to vesicles, to like a population of vesicles. And then we ask, if we encapsulate RNA inside the vesicles, does that have any effect on the RNA activity? So again, we started out looking at the malachite green aptomer. This aptomer causes the malachite green dye to fluoresce upon binding. Also, another important point was that malachite green, the dye, is permeable across the membrane, so we can make equilibrium measurements. So our question is, how does encapsulation affect binding affinity? And we do this by comparing two scenarios. The scenario where there are vesicles, but the malachite green aptamer is not encapsulated. So we call this like an outside vesicle condition. And then also the condition where the RNA is encapsulated inside the vesicles. So what we see is that if you compare the situation inside versus outside, here we're looking at a binding curve. As you increase the amount of malachite green, you get more fluorescence. This binding curve is shifted to the left, indicating more binding at lower concentration, lower KD, when the aptamer is encapsulated. And we also see similar results for phospholipids, POPC and DOPG, different phospholipids. So the nature of the membrane, it doesn't have to be a fatty acid, at least. It seems to be relatively tolerant to different lipids. Having said that, if you use a positively charged lipid, then that'll precipitate with the RNA. And so we can't really get a good encapsulation experiment there, but at least for these negatively charged phospholipids, they appear to behave similarly to the fatty acid vesicles, which are also negatively charged, or zwitterionic, I guess. POPC is zwitterionic.
Is this because the this enzyme, the ribozyme inside the vesicle effective concentration is high?
So we think that it's an effect on the folding. So let me kind of address this in a couple of ways. So one is that the dye should be at equilibrium because it's permeable through the membrane. So we're trying to make equilibrium measurements. The other thing is that we have done some experiments that I didn't prepare any slides on, but most recently we've been working on the hairpin ribozyme. And the hairpin ribozyme has, I guess there are a lot of constructs that people have made that we could take advantage of. One of them is a fret labeled construct where one of the reactive intermediates along the reaction pathway is a docked conformation. So there's kind of like two arms, I guess, of the hairpin, and then the docking causes these two arms to come together, and that is a necessary intermediate on the reaction pathway. So we can monitor fret of that docking interaction. And what we see is that if the hairpin ribozyme is encapsulated, then docking is promoted. So we think that there's some folding effect based on these two things. We think that the folding is stabilized.
But the 26 nanometer is quite large, right? I mean, yeah,
it is pretty big compared to the aptamer. If we could make these vesicles smaller in a controlled way, then yeah, I would think that you get a better effect from that. We have gone in the opposite direction, looking at larger vesicles, but that's heterogeneous. It does get worse, but you can't really be quantitative because it's a heterogeneous population when you go a little bit larger. The problem with going smaller is that the vesicles are not stable at the small sizes for very long. They tend to fuse. And with these equilibration experiments, we incubate them overnight. So we couldn't really get a good population of vesicles that way either. Yeah, so if there was a way to make unilamellar vesicles at like multiple sizes that we could work with here, I think that would be really helpful for pinning this down. Okay, so yeah, we were thinking that encapsulation stabilizes local interactions. I think there's the study I just told you about with the hairpin ribozyme, where I think we looked at it specifically. Here it's a little bit indirect, but we look at the melting temperature and the, so it's the fluorescence of the aptamer dye complex as the temperature increases. And if it's encapsulated, then we see that this transition temperature is shifted to the right, or it occurs at a higher temperature, meaning that this, at least the binding has been stabilized. So a couple other things that we looked at were that there is a shift if we look at shape, which is a mechanism for probing the secondary structure. There is a shift in the secondary structure toward the bound form when the ribozymes are, I'm sorry, the aptomer in this case, when the aptomer is encapsulated. So that is also consistent with kind of the conformation shifting toward the active form. And then we also, as I said before, we tested a few different vesicle types. We see a similar result. And also in some crowded conditions, we get this same shift. I have to say that with crowding agents, it's a more complicated story. I think actually even more complicated than the vesicles, because crowding agents can have some significant chemical interactions with the RNA. So depending on what the crowding agent was, we would get different effects. So some of them really denature the RNA. Some of them seem to, crowd the RNA and improve activity. So whether you use PEG or FICOL or BSA or, what you choose, I feel for the crowding agents, we would see some diversity there. So if anything, this kind of, vesicle encapsulation seemed to be a little bit more consistent when we varied the vesicle types. So to understand what was going on, we considered this to be consistent with the expectation of some theorists. Zoe and Dill had created this model of a confined biopolymer in a spherical confinement. And the general idea, if I understand it, was that we have this biopolymer, which can exist in a variety of different conformational states. Some of these states are more extended and some of them are less extended. And if you have an encapsulation, especially if the encapsulation is on the smaller end, then that disfavors these extended states and you get a relative stabilization of the compacted state. So if the compacted states are also the folded states, then confinement can shift the equilibrium of these structures toward the active form. So this lines up with what you were saying about if we could make these smaller, then that could improve them. Or if we make the RNAs bigger, maybe that could also have a similar effect. And this is just illustrating what we think is happening. The vesicle membranes are kind of very primitive kind of chaperone for the RNA and encouraging them to adopt a compacted structure. And again, if the RNA has an unfolded form and a folded form and it's encapsulated, then this destabilizes these extended forms and gives a relative stabilization to the folded form. Okay, so that was with malachite green aptomer. We kind of made some observations that were suggestive of there being some, kind of chaperoning or folding benefit to being encapsulated, and that's consistent with the expectations based on biopolymer models. Then we decided to ask ourselves, what would happen if we encapsulated ribozymes and evolved them? Would there be something different about the way that they evolved if they're encapsulated in these vesicles versus not? So to look at this, we encapsulated the ribozymes that I told you about previously, the self-amino-acylating RNAs. We know that the oxazolone substrate is permeable through the membrane, so it's not a problem to conduct this reaction inside the vesicles. And then we also have an outside vesicle condition where we evolve the RNA in the presence of vesicles, but not encapsulated. So here's an example of just looking at an isolated ribozyme, very analogous to looking at the malachite green aptomer. In this case, of the isolated ribozyme, we ask what's its activity. We can measure the reactant fraction over time in the assays that I've talked about previously. And we can see that for this ribozyme, the encapsulated ribozyme gives higher activity than the ribozyme outside the vesicles. So that's consistent with what we understand. Then we did kinetic sequencing to look at this for, I think it was some thousands of ribozymes. I'm forgetting the exact number, but about 5,000 different sequences. And these sequences represent the different motifs that I told you about previously, motif 1, a couple of sequences, a couple of the subfamilies from there, the 2 subfamilies from motif 2, and then there's only 1 family in motif 3. So we measured the activity of the different sequences and plotted the activity when they were encapsulated, which is along this axis, the y-axis, versus when they were outside the vesicles on the x-axis. So the first thing you notice is that, just like with that one sequence I showed you earlier, the points tend to be above the line of unity. So there's a tendency to have higher activity when the ribozymes are encapsulated versus when they're outside the vesicles. Maybe that's just consistent with everything we've been talking about. But I think what's a fascinating property of this plot is that if we focus in on the high activity sequences, there is a deviation, a greater deviation from the x equals y line. So the high activity sequences benefit more from encapsulation compared to the low activity sequences. So taken together here, if you were to fit all of this data together, and these points down here are less active sequences, and natural selection is more weighted toward or more concerned with the high activity sequences. So if we plot the line of best fit, it is actually kind of tilting in this direction relative to the x equals y line. So this seems kind of subtle, but it was fascinating to us because this says that there's greater variance among sequences when they're encapsulated. And in Fisher's fundamental theorem of natural selection, the rate of adaptation, the rate of evolutionary adaptation, is proportional to the variance. So if you have a very highly variable population, then adaptation is faster because the high fitness sequences quickly crowd out the low fitness sequences. Whereas if everything is very close in fitness, you have many individuals with similar fitness, then adaptation moves very slowly because there's just not that much variation in the population. So our prediction is that if we conduct in vitro selection inside vesicles, we should get faster adaptation or faster convergence onto the most active sequences when the ribozymes are encapsulated. And that's what we see. So here is a plot of the diversity index. I think this is the Simpson index. It's the probability that if you pick two sequences, the probability that they are different. So when this Simpson index decreases, that means that the population is converging onto the ultimate solutions or the high activity sequences. So with the encapsulated encapsulated selection, this happens at a particular time. Around round 2, we start to see convergence. When we do this selection outside vesicles, then we see convergence later in round 3 or 4. So given how short this selection is, this shortening it by 1 or 2 rounds is a pretty significant acceleration in convergence of the population. So, if I could summarize that project, it says that encapsulation helps all sequences. So in general, we saw everything kind of above the x equals y line, but it helps the active sequences more than it helps the less active sequences. So this is kind of a Matthew effect, or kind of a rich get richer effect, which gives you faster adaptation through natural selection. So what's the basis for this, or how do we understand, like, why are the active sequences helped more by encapsulation? Because, going into this, my intuition was actually that, okay, the active sequences are already well folded. They don't need much more help. Whereas encapsulation could really help those sequences that are kind of borderline, and those sequences could get a big boost from encapsulation. So I thought that it would actually decrease the variation among sequences. So what I think was wrong with that intuition, that initial intuition, what I think is wrong with that is it assumes that you have some well-adapted sequences. So here I'm just plotting fraction folded against melting temperature, kind of like a postulated melting curve for these ribozymes. That thinking that you can't really help those good sequences more, they're already well folded, that's kind of assuming that we have sequences that live up here on this curve. And yeah, if you increase the fraction folded, they there's not that much headroom. On the other hand, probably where we actually are now that we think about it is these are short sequences. They're not incredibly active. These sequences might be more around here in fraction folded. So they are on this part of the curve where a small change in stability could give you a small change if you're at a lower position. But if you're already at a higher position and you get the same small bump, then maybe you get a higher increase in the activity through the fraction folded. So, this is a very rough diagram, but I think that is kind of my intuition right now for why the active sequences benefit more from encapsulation. Okay, so I'm going to switch topics again. I'm happy to answer any questions about what I've just talked about right now, if anybody has any questions. Okay, if not, I'll just go ahead and talk about the last topic here, which is an application. And it might not be obvious from the outset, but what I'd like to convey to you is that the evolutionary robustness or robustness that was developed during evolution can be useful for biomolecular engineering.
Sorry, I just before you start, I did get a question.
Oh yeah.
So it's a cool hypothesis, and I guess what I would be interested in is there a way of more directly testing how well folded the sequences are?
Yeah, so we have thought about trying to do it in a high throughput manner. We have not executed any of those plans yet. Yeah, one could, I guess brute force would be to, individually measure the melting curves for the different ribozymes. But yeah, I think that would be a really interesting question to answer.
Cool. Cheers.
Okay, so I'll go ahead. Okay, so we've been talking about kind of systems for early life and the emergence of life. And now I'd like to kind of fast forward about 4 billion years to a system which has experienced 4 billion years of fast adaptation. And that's the evolutionary arms race between hosts and pathogens. So I just want to introduce this idea quickly if you haven't heard of it before. The evolutionary arms race is kind of this idea that we're seeing this kind of in real time also, that pathogens evolve to infect hosts, hosts evolve to resist pathogens, and then those pathogens counter-evolve to be able to reinfect those hosts, and then you kind of get this, this race of needing to evolve as fast as you can in order to avoid getting outcompeted by the host, either the host if you're the parasite, or by the parasite if you're the host. So this is called a coevolutionary arms race. It's also called the Red Queen effect. And this, this name goes back to Alice in Wonderland and Through the Looking Glass, where the Red Queen is pulling Alice through this landscape, and they're both dashing along as fast as they can. And at some point, Alice looks around and she notices that the scenery isn't changing. She's actually staying in the same place. She's running as fast as she can, but she's just in the same place. And Alice turns to the Queen and she says, well, in our country, you generally get somewhere else if you run very fast for a long time, as we've been doing. And the Queen says, a slow sort of country. Now here, it takes all the running you can do to keep in the same place. So this is what hosts and pathogens are doing, evolving as quickly as possible just to stay alive, just to stay in the race. So perhaps the longest running Red Queen race is that between bacteria and viruses that infect bacteria. So we've had at least 4 billion years of this. So these viruses that infect bacteria are called phages, And phages are everywhere. They are the most abundant organism on the planet. The estimation is that there's about 10 times more phages than bacteria in terms of numbers. They recognize and kill bacteria. So that's perhaps of obvious utility. Perhaps we can use them for therapy. People have thought about that for more than 100 years now. Basically, ever since phages were discovered, people have thought, can we use them to kill bacteria? And also, something I'd like to keep in the back of our minds here is that the proteins that are involved in host interaction experience a high evolutionary rate. So receptor binding proteins, for example, that bind to the host receptor, they evolve quickly. The host receptors are also trying to evolve, to avoid predation. And more importantly, the phages are evolving to continue to be able to infect their chosen hosts. Okay, so what are some problems with phage therapy? Because that's kind of what we're looking toward. This idea, it's been around for a long time. The thought that maybe somebody with a bacterial infection that's not responding to antibiotics could be treated with phages. And there's been some high-profile anecdotal experience on this, where individuals have been, exhausted all of their antibiotic possibilities, and somebody's tried a phage and it has worked. So what are the problems with phage therapy, though, and why isn't it widespread right now? Most phages are poorly characterized. So the way that phage therapy is typically done is that there's a bank of phages. You have a strain of bacteria, you look in your bank and you ask, does this phage infect the bacteria? If it does, then I'll just prepare some of this phage and inject it into the patient. This is fundamentally problematic because most phages are poorly characterized. And there's a real risk of genetic transduction from phages, meaning transfer of undesirable genes by the phages. There's a documented transmission of antibiotic resistance genes among Staphylococcus aureus by phages, for example. Some phages are actually detrimental. They help form biofilms with Pseudomonas aeruginosa. So there are downsides to using a phage that you just don't know that much about. Phages amplify exponentially. Sometimes you think of this as a positive because you only have to give a small dose, and if the phage infection takes off, then you don't need to supply more. The flip side of this is that it's very difficult to dose. What if you didn't give enough? It just won't work. It'll be kind of all or nothing. It can be difficult to get repeatable results in studies. So this is exponential amplification. Even though it seems nice, it might not necessarily always be desirable if you want to have a well-controlled therapy. And of course, there's the possible risks of using an evolvable organism. There's open-ended evolution. We kind of just would prefer to avoid that uncertainty. So all of these things mean that phage therapy, as it's kind of like typically envisioned, can give some unknown biological consequences. So what we'd really like to do is to make phages more drug-like. So we want to take advantage of the things that phages are good for, such as finding bacteria, binding to bacteria, and then maybe use a nanomaterial to destroy the bacteria as well as the phage, so that we can avoid some of these downstream consequences that are kind of unknown. So to try to control this effect, we've been making phage nanomaterial conjugates. This is a kind of artist depiction of this. We're using M13, which is a very well understood phage that normally infects E. coli. It's a filamentous phage, a rod-like structure phage. At one end of the rod is the receptor binding protein. This is the part that the protein of the phage that binds to the bacterial receptor. So this is a place where we can engineer it in order to bind to a particular pathogen that we're interested in. This M13 is sort of serving as a scaffold where we can attach cargo. And the cargo I'll tell you about is gold nanorods or gold nanoparticles for different applications that I'll explain. Okay, so focusing on the targeting, We have M13 and its receptor binding proteins, the wild type, which binds the F. pilus of E. coli. We can swap this out with receptor binding proteins from other phages. And there are now thousands of putative phages belonging to this family of filamentous phages in a database. But not too many of them have been explored in detail or experimented on. This is kind of like for metagenomic studies, but still we can pick out a few that have a known interaction with an interesting host. PF1 is something like that. It's a phage that binds to Pseudomonas aeruginosa. So we can basically just cut and paste its receptor binding protein, express it on M13. Now, in order to attach the cargo, or gold nanoparticles and gold nanorods, we treat it with EDC and this reagent, which will thiolate the phages. So the thiols, this is a pretty non-specific reaction. The thiols on the phage will then be able to interact with the gold. So our proof of concept here was with gold nanoparticles. These are spheres, and gold nanoparticles and also gold nanorods exhibit something called surface plasmon resonance, which is a collective oscillation of the electron cloud, which occurs upon absorption of electromagnetic radiation at the right wavelength. So if for nanoparticles of this size, roughly 4 nanometers, then we get absorption such that it appears to be a red color. So this absorption occurs at a particular wavelength, a resonant wavelength, and therefore changes in this absorption, which are caused by changes in the surface properties like aggregation or changes in the size of the nanoparticle, those will affect the color of the nanoparticles. And this is a picture I got from Wikipedia of colloidal gold being used in glass to color it red, cranberry glass. So again, because this is a property of the electron cloud, it's quite sensitive to the aggregation state of the nanoparticles. So if they are dispersed in solution, they're red. If they're clumped together, then we get a different absorption, which is purple. And that's it's being taken advantage of in different fields, particularly in cancer nanomedicine for detection. So in this proof of concept experiment, what we did was to mix our phages, which have been retargeted to Pseudomonas aeruginosa, a human pathogen, mix it with the bacteria. We get the phages bound to the bacteria. We can spin these down. Then we add our gold nanoparticles and they aggregate on the phages and give a color change. So here's what that looks like. This is a detection of about 100 cells for this kind of red to purple transition. It can also be seen on the absorption spectrum. And this is robust to different environments. We've done this detection in tap water and seawater, human serum, milk, urine, a porcine ex vivo model. This is basically a pig lung that Pseudomonas aeruginosa is grown onto. So it's this assay tolerates these different conditions. And I think what's very interesting is that we engineered the specificity of the phages to be E. coli specific, or Pseudomonas aeruginosa specific, or Vibrio cholerae, or Xanthomonas campestris, these different species. And we just did basically cutting and pasting with the receptor binding protein sequences. No evolution, no optimization, which was a little bit disappointing, because we like to do evolution. But still, just the fact that these worked without any optimization was something that kind of stuck in our minds. Okay, so now we wanted to think about an application that could be interesting going back to phage therapy. I just want to make the point that antibiotic-resistant infections threaten many aspects of modern medicine, not just infections as a primary disease. So we certainly think about, what it would be like if we didn't have antibiotics to treat pneumonia or urinary tract infections or whatever. But not only would we lose that capability, if our antibiotics stop being as effective, but also other parts of modern medicine, like surgeries, very routine procedures, suddenly become much more dangerous because we can't guarantee that the patient won't suffer a life-threatening infection as a result of the procedure. So a surgery, chemotherapy, these people are immunosuppressed, and we rely on antibiotics to take care of their infections. And there are many, many conditions like this. So really, I like to think of antibiotics not only as treating infectious disease, but as kind of a foundational piece of modern medicine that many other parts are built on top of. Okay, anyway, so I think we all acknowledge that antibiotic-resistant infections are an important problem to try to solve probably within our lifetimes. So what modification can we make to our strategy in order to kill the bacteria, not just bind to them? I mentioned how the colloidal gold absorbs due to surface plasmon resonance. The flip side of that, so there's an absorption, which we can see as a color change or as a color. The flip side of that is what happens to the absorbed energy. So the absorbed energy results in electronic excitation, and that excitation is then translated into heat or vibrations in the gold lattice, and that's released as heat. And this is called photothermal heating or plasmonic heating. And that effect has been used, is starting to be used in cancer nanomedicine to kill cancer cells. So we thought we would use the same technology to try to kill bacterial cells by coupling these gold nanoparticles, nanorods now, to phages. Now, the reason why we're using nanorods, slightly different morphology, is because we can tune the shape of the nanorods to absorb in the near infrared. And in the near infrared wavelengths, biological tissue is somewhat transparent, at least to the depth of a few centimeters. Okay, so we have our nanorods. Normally, they do not interact with bacteria. This is a bacterial cell. These are the gold nanorods. But if we attach them to phages, then these phages bring the gold nanorods to the bacteria. And you could see that if we were to activate the gold nanorods here, then they would heat up right next to the bacteria. And this works pretty well. So here we're looking at destruction of bacteria over time for different types of bacteria using our different engineered phages. This is in vitro here. And we kind of followed up on Pseudomonas aeruginosa because it's a major pathogen. 18% of clinical isolates are multi-drug resistant. And what kind of is alarming, I think, is that resistance actually emerges during antibiotic treatment in 10% of infections. So the CDC is concerned with this. The WHO calls Pseudomonas aeruginosa a priority 1 organism. So this is one of the species that we'd like to tackle. Okay, so in the context of mammalian cells, one question we want to ask is, if we have bacterial cells growing on mammalian cells, like a biofilm of cells on the mammalian cells, can we kill the bacterial cells without harming the mammalian cells? Because we are creating kind of a thermal field. Is that just going to ablate all of the cells in the petri dish? So here we have a Pseudomonas aeruginosa biofilm being grown on mammalian cells. This is red-green staining, where green is a dye that penetrates all cells, and red is a dye that only penetrates dead cells. So we can see before treatment, before irradiation of the phage nanorods, there are basically no dead cells. You see the little ones are bacteria, and these large ones are the mammalian cells. But then after irradiation, you can see there's widespread death of the bacterial cells, and mammalian cells do survive. And it's a little hard to see it here, but we have more quantitative assays for this, and the mammalian cells are largely intact. And then also, no replicating phage remains after treatment. And this was one of the reasons why we were interested in this strategy in the first place, was to prevent any downstream problems with the phage. I want to make one point about phages versus antibodies, because often people say, well, all you did was use the phages to bring your nanorods to the bacteria. Why can't you just use antibodies to bring your nanorods to the bacteria? So there's some theoretical reasons, some conceptual reasons. One is that you'll get with phages greater delivery per binding interaction because the phages are much larger than the antibodies. So our phages are carrying maybe on the order of 15 nanorods per phage, whereas an antibody would probably just have one, maximum two, just because of its size. And this translates into greater efficiency of cell killing, and that's seen in vitro. Here comparing phage nanorods with antibody nanorods. But I'm going to show you some in vivo data which shows the same thing, and that's maybe a little bit more compelling. Okay, so we know that we can, treat bacteria in vitro. What about in vivo? Because there are a lot of things that can go wrong in vivo because of the complexity of the system, immunological responses or whatever. So the model that we used is mice, where we make a wound on the back of the mouse, and then infect that wound with Pseudomonas aeruginosa. Then we wait anywhere from doing treatment on the same day, so just waiting a couple hours, or waiting up to a couple of days. We wait for that wound to mature. So then we treat with the phage nanorods and we irradiate. That's done on the same day. And we do it 2 days in a row for most of the experiments I'll show you. We also made a version, which I won't really go into the details, but we made a version that also releases zinc. And zinc is kind of an ion that promotes wound healing. But we treat with the phage nanorods and then follow wound healing over time. So first, let's look at the top panel here, just illustrating that if you have a control, which is just PBS, if you irradiate PBS, there's no change in the temperature of the mouse. This is a thermal camera. But if you treat with the phage nanorods and you irradiate, then you see an increase in the temperature of the wound. And we optimize this so that the temperature is high enough to kill the bacteria, but doesn't leave a burn on the wound. Here's the natural history of a wound over time. So you can see here's the initial incision, and then you can see a progression of the wound. It gets worse acutely, and then it starts to improve slowly. So wound size over time for this control, no treatment control, wound size increases, and then it decreases as the mouse kind of heals, catches up to that infection. However, if we treat with the phanorods, and this treatment was done quite early, so it was done just a couple of hours after infection, then we can see there's a very good healing, and you can see that in the wound size here as well. If you look at the histology, this might be, it's a little bit beyond me. It might be kind of beyond what you're used to seeing, but in this control, all of these little purple dots are neutrophils, which are indicative, this like huge kind of collection of neutrophils, a huge density of neutrophils is inflammatory infiltrate, indicating a inflammatory response from the mouse. But you can see that in the case of the treated wounds, we don't have that kind of huge inflammation. And we approach that in day 10 of the control, but we get that very quickly with the treatment. One thing we want to know immediately is how do these phage nanorods compare to standard treatments? In the case of Pseudomonas aeruginosa, that would be ciprofloxacin or levofloxacin as a standard treatments for these infections. So we also treated the mouse with Cipro and Levo, which are these two lines here, blue and purple, and also some other topical antiseptics, which are probably not a very common treatment for humans. But I think what you can see is that antibiotic treatment is actually a little bit slower than the phage nanorod treatment. And you can see one of the metrics of that here, where if we look at the bacterial count, in the case of the antibiotics, purple and blue here, the bacterial count does go down, but it takes a little time to get there. So there's a lag between the application of the treatment and the effect of reducing bacterial load. Whereas with the phage nanorods, it's a targeted treatment right at the wound. It does give you results quickly in terms of reducing the bacterial count. Okay, what about a situation where, most people, they get a wound, maybe it gets infected, but they don't realize it's a problem until a couple of days later when the wound's gotten bigger and they're feeling sick. Same thing with a mouse. The mouse wound increases in size for a maximum around 2 to 4 days. So what if we delay treatment until day 2 and ask how well the phage nanorods work? What you see is that they work pretty well, starting again just from that time of treatment. You get good improvement already. Whereas for ciprofloxacin, for example, it takes a little time for the ciprofloxacin, a systemic treatment, it takes a little time for it to act on the wound. Okay, now what about a really bad wound where it's a larger wound and you infected it with a higher amount of Pseudomonas aeruginosa so that it would be lethal to the mice if untreated. So you can see in the control, these mice don't live past day 3. So this is, I guess, a situation where treatment is required. In the case of the phage nanorods, it's possible to go from this scenario where really close to point of no return and have a good result from that. Of course, if you give the treatment early, then you can also have a very quick result from that. Okay, I think the last mouse experiment I want to tell you about is we wanted to try to mimic a scenario where we had a strain of Pseudomonas aeruginosa that was resistant to antibiotics, the typical antibiotics that would be used. So it's the clinical treatment of Pseudomonas aeruginosa starts usually with ciprofloxacin and levofloxacin or maybe some other agents. If that fails, then there are some novel agents that can be used, such as combination beta-lactam with a beta-lactamase inhibitor. So there's some of these, newer agents that could be tried. And then if that fails, the last option is polymyxins. So polymyxins like colistin or polymyxin B are a kind of last resort. One of the big problems with them is that they're very nephrotoxic. So in the majority of patients who are given colistin or polymyxin will have some renal failure. So to mimic a scenario where polymyxin and these other agents had failed, we used a polymyxin-resistant strain of Pseudomonas aeruginosa to inoculate these wounds. You can see that as expected, the polymyxin line, this blue line, is basically the same as the control. There's no effect from treating the mouse with polymyxin. It's a resistant strain. Whereas if we treat with the phage nanorods, if we do irradiation on day 2, or if we start irradiation immediately, we can still get, in both cases, we can get healing of these wounds. Oh, okay. I do have one more slide on this topic. So we also asked about the scenario where perhaps your patient couldn't stay in the hospital and undergo 2 treatments. What about just 1 treatment? Or what if you just dosed them once with the phage nanorods? Would that be sufficient to help heal this infection? What you see here is the control again, and in purple is a single dose. Pink is a single dose. In red is that double dose, which is our kind of normal regimen for the mouse. So you can see that it's not as good as having 2 doses, but it's not terrible. And actually, it's about the same as if the wound had not been infected. So healing from a single dose it brings you to about the same as if there had been no infection to begin with. So the question that people always ask with a new therapy is, okay, maybe it works to kill the bacteria, but what about the toxicity on the animal? And we've done some histology to show that there's no obvious effect on the kidney and the liver, which are the places where you tend to see toxicity. You can follow biomarkers also to look at toxicity. Here we're looking at, on the top row, some markers of liver function, and the bottom row is some markers of kidney function. And the antibiotics shown in blue here they all give elevations of these biomarkers, and that's consistent with their known toxicity. It's a systemic treatment, whereas the phage nanorods is a localized treatment. We're not expecting toxicity in the kidney or the liver, and indeed that's we don't see any toxicity, which is consistent with that localized treatment. Okay, sorry, I know I keep saying this is the last slide, but then there's like one more. So here, this is a comparison of phages versus antibodies for targeting. So antibodies are very, popular, and they're in already many biologics. So why not just use those? Maybe they're not bad. They do result in wound healing compared to the controls. But the phages appear to do better, and I do think that's because they're simply carrying more cargo per interaction to the bacteria. So I guess the possible clinical scenario that I'd like you to sort of entertain is that maybe you have a patient with a wound infection and antibiotic treatment has failed. Then we'll take a sample of that, send it to the lab, check whether or not we have phages that can bind. And we can do that quickly with the phage nanoparticle assay, which takes maybe 30 minutes or so in the clinical lab. And then if you confirm that there is susceptibility, that there's binding, then you can attempt this treatment with the phage nanorods. Okay, so now I want to kind of try to bring it back to what we were talking about with evolutionary robustness. So one thing that we noticed and that stuck in the back of our minds was that we didn't have to do any special optimization or engineering to make these chimeras of M13. So we took M13, an E. coli phage, and just basically pasted on the receptor binding proteins from phages that have no obvious homology to M13. We know that they're in the same family because they have the same genome organization. But if you were to align the receptor binding proteins, then you would be hard pressed to find a real alignment. But because we know that these are related phages, even though they're highly diverged, we did this cutting and pasting, and it turned out that we didn't have to do any optimization. So why is this? Not only did we do the swapping, but we also did chemical modification. We decorated them with nanoparticles. We put them in some weird environments. The phages were kind of fine with all of this. And we kind of wonder why. We obviously don't know the answer, but I'd like to speculate that perhaps the Red Queen effect has something to do with this. So rapid evolution, which is caused by the Red Queen effect, means that phage proteins have been evolving, and these receptor binding proteins in particular have been evolving quickly, as fast as they can. And that means that the scaffold that they're built on must be robust to mutations. Perhaps the robustness to mutations that is the result of the Red Queen effect means there's some overlap between being robust to the modifications that are mutations, genetic mutations, and the modifications that are chemical in nature that we think of as being chemical modifications. So robustness to evolution perhaps is analogous to robustness to engineering, and that means that we can look in these places where we see the Red Queen effect to try to draw out proteins that will be easy to work with for engineering purposes. So I'd like to thank all of the members of my lab who have been working on these projects, especially the ones highlighted here have been involved in the projects that I talked about today. And I also want to give big thanks to our collaborators, especially Ziwei, Ken, and Maria, who again were involved in the projects that I discussed today. Thank you very much. I'm happy to answer any more questions.
Okay, thank you very much. So now it's a time for asking some question from audience. Anybody has a question? Let's see. So I have one, maybe just a naive question. So you, in the first part of our talk, you are describing this amino acylating ribozyme, And how big is this lysine in terms of, sequence length? And then you mentioned that you are doing this directive evolution. So are you really exploring all the sequence space, like 4 to the n in the experimentally? Is it possible? And the next question is, how do you actually do this selection in the experiment, in practice?
Okay, so this is an interesting question. The setup of the, actually I can go to the slide that shows the structure of the library. So here, this is a random region. So shown in blue here is a random region. So here we do have N21. And for the 21, we can explore that in sequence space. At least we can synthesize a library that contains maybe roughly 1,000-fold coverage of a 4 to the 21 sequence space. These black regions are constant regions, so they are invariant. So they likely participate in the ribozyme. This is something that is kind of part of most selections because they're PCR handles. You need some sequence for amplification, but they have kind of an unknown effect on the outcome. So, if we wanted to go deeper into this, we would try different. We would try different flanking regions here to see how if they change the overall outcome. It's likely that if you change those flanking regions, because they are part of the secondary structure, it's likely that if you change them a lot, that you would get different ribozymes. And whether the overall picture that you have a few different motifs and the network, the evolutionary network is frustrated, whether that is the same, I can't say. I would suspect that that kind of general message might survive that change here in the experiment, but I don't know for sure.
Okay, thanks. So anybody else?
I've got a question.
Yes.
Or two rather. So first, so you can, it's not very computationally expensive to calculate what's the secondary structure of an RNA of this size. And I was wondering, have you looked at what is the predicted secondary structure and also energies of these structures?
Oh, yeah, we did just kind of, run them through RNA fold or Vienna fold, I guess. And I don't have a slide here with that. It is in the supplementary material of our paper. I'm not sure if I kind of learned anything from the secondary structures. We also didn't validate them. So we would want to do some, biochemical probing or some other experiments to validate the secondary structures. So that's kind of why, I don't put too much emphasis on them because I guess they're, a prediction. And
not just the sequences that you isolated, but also the sequences that didn't get. Do you have any information on the sequences that didn't get selected? And I
guess all I could say about those sequences is that most of them were in the starting pool, but they presumably didn't have that much activity. We did do a duplicate of this selection. We tried to do it twice independently to see whether or not we missed anything big. I think that the sort of islands of activity are large enough that even if a single sequence drops out, it's unlikely that you'll miss an entire motif. So yeah, I guess but I don't have any. I don't have any insight into what got left behind.
Mm-hmm. So my second question is, so your neutral network was defined by, but just by things that worked, by things that were selected. And I was, so that's kind of like a binary sort of a view of a neutral network. Either you're in or you're out. But of course, the different sequences will have different sort of catalytic activities. I was wondering if you looked at the landscape in terms of that also.
Okay, so that information is we tried to capture that here.
Yeah.
We kind of said, below an activity of 5. So here the catalytic ratio is the ratio of activity that's catalyzed to the background reaction rate. So there is some background rate, and so these are expressed as the ratio. We said that if it's below 5, and that's maybe a little bit arbitrary, but we felt that it was if the catalytic activity we calculated or that we measured was 5 times higher than the background rate, that might not be statistically significant. So that was kind of like the basis of our cutoff. So yeah, this is not showing all of the functional sequences. You'd have to go into the attached data file to see all of them, but this is more showing the result of a pathfinding algorithm. So it might only be like less than 100 sequences plotted here, which are paths between the 5 or 6 larger dots. And that was the way that we used to visualize this. There are probably better ways to try to visualize the data, but what we're trying to get at here is looking for these pathways.
Very cool. Yeah, I missed that. Thanks.
Any other question? Let me ask one more question. So why did you choose this amino acid lating ribozyme as your model system? I mean, in 1980s, the Tom Check, he for the first time he showed that this T ribozyme, tetrahymena ribozyme, group 1 intron has catalytic activity. That was the kind of groundbreaking work, right? So, and then you can actually, is there any reason you chose this particular system instead of ribozyme?
Yeah, definitely. So one of the reasons was because of its, being interesting for the genetic code. But then the other reason, which was equally important, was that some of the ribozymes people had found for this particular activity, for the adenylation activity, were very small. So we wanted to kind of improve our chances of finding ribozymes by looking for activities that appear to be kind of doable by small RNAs. And I don't know if you saw this paper by Michael Yaris's group on the minuscule ribozyme. It was a 5 nucleotide ribozyme, I guess you could say, but I mean, the substrate was also it also had an RNA component, 5 to 13 nucleotides, which catalyzed amino acylation. So I think that was kind of one of the things that said, okay, well, this activity, we can probably get something out of a selection if we do it, because the greatest risk to doing a selection is getting nothing. So we wanted to try to increase our chances of finding something, even though the random range is pretty short.
Okay, so looks like we had a nice discussion, and then okay, let's thank Irene Chen for her wonderful talk. Okay.
Thank you.
Thank you very much.